Okay, this is the third lecture of Introduction to Behavioral Neuroscience B. Today's lecture is about learning in neuroscience. Uh, I will upload the video, and also this weekend I'm going to put up the first two quizzes about Lecture 2, Lecture 3, and I'll send an email to all of you so you know where to find those. Don't worry about those yet. You'll have 2 weeks to do them after I upload them, and I'm pretty flexible, so I'll probably give you a little bit more time. Today, there's basically four topics I'm going to cover. One is forms of learning, so I'm going to go over sort of the different categories of learning. Then I'll talk about a specific theory of how learning happens in the brain called Hebbian learning. To understand Hebbian learning, you really need to understand sort of basic artificial neural network theory. So I'll give a quick introduction to neural networks. And then I'll talk about how neural networks might be implemented in the brain. So I'll talk about synaptic plasticity, two types, LTP and LTD, long-term potentiation and long-term depression. Why is learning important? Well, humans are really good at adapting and learning. Humans, moved all over the world to many different climates very quickly over the period of only a couple tens of thousands of years. And in each, of course, location, they encountered different types of plants, different animals, different climates to survive. And you can see, different cultures throughout history have had very different solutions to survival, where humans have pretty much thrived everywhere, wherever they've gone. So quick adaptation was crucial. You didn't have time, like some animal, to sort of slowly, over thousands or millions of years, to change your natural fur and all of this stuff. You had to have basically learning within individuals and sort of within cultures, societies, and learning is and adaptability is what adaptability after birth. So non-genetic adaptability is what enabled that one of the things that enabled that quick spread. Another reason that learning is really important is, for example, for humans, right now we're in an interglacial period in the Earth's history that began around 11,000 years ago. The last glacial period lasted about 100,000 years, between 110,000 years ago and 11,000 years ago, and the peak of the glacial period was 20,000 years ago. So it was really cold, and there was lots of glaciers, and there was more land because the seas were more frozen. So what I want to say is that basically the environment can change rapidly in just a few generations. In 9,000 years, over a couple hundred generations, the environment changed significantly from having land between, for example, the United Kingdom and France and Germany, to having no land there. And so quick adaptation to the new climate and environment was necessary for survival. And so people who had better ability to learn were probably able to more quickly adapt and respond to the new environment. Anyways, that's sort of the just so evolutionary argument for why learning is important and how it helps. So now, next I'll go into the second topic, which is forms of learning and memory. In general, sort of psychologists have separated memory into two major sections, two major types. There's declarative memory, which is memory sort of about things, about the world. It includes things like semantic memory, like facts, etc. Paris is the capital of France, and I left my, glasses are made out. Glasses can break. Episodic memory is more about life episodes, sort of memory. Like I remember I put my cup over on the table, or I remember I learned how to drive when I was, learning to drive when I was 16 years old, or something like this. That's declarative memory. There's also sort of procedural memory in between. I guess it's non-declarative memory. The other type of memory is non-declarative memory, which includes sort of three different subsections usually, which is one is motor learning, which is learning to catch a baseball or something like that. And the next is association learning, associative learning. And the other one is aptly named non-associative learning, which is everything else. And in neuroscience, in this class, we're especially going to investigate today. We're going to talk about these two types of learning because they're very basic, sort of automatic types of learning and memory. So it's a good place to start to understand the brain. Within non-associative learning, you have two main types we're going to talk about. One is habituation learning. The other is a phenomenon called sensitization. Within association learning, we're going to talk about two types. One is classical conditioning, sort of Pavlov's dog, if you know about this. And the next is instrumental or operant conditioning. Okay, so we're going to start off with habituation. So habituation is the reduction of a behavioral response to repeated presentation of some stimulus that has no meaning. So for example, the first time you hear a noise from outside, like the hammer from a construction site, you might jump and look over it and try to see what's wrong. But then after they keep hammering over a long time, minutes or hours or days or whatever, your response will decrease over time. You'll start to be less surprised by the hammer until sometimes you even ignore it totally. Your response goes to zero. From neuroscience point of view, habituation was mainly the neural circuits of habituation were investigated in what's called the aplysia, which is a sea hare, a Californian sea hare. And this is a sort of sea slug animal in the ocean that when you stimulate its gill, when you shoot some water or some air or something at this, then the contracts the gill. The gill contracts when you put water on the siphon, like to represent some stimulus, something touching it, right? And then if you keep doing that, then the gill retracts less. So this is before you stimulate this, and then this thing that's absorbing air from the water, oxygen from the water, it gets smaller because it doesn't, it's scared, it doesn't want to have its gill eaten or something. But if you keep doing it, then it retracts less until it doesn't retract at all. This was studied by Eric Kandel, one of the main author of the Kandel Neuroscience book, one of the most commonly used books in graduate high-level neuroscience courses. Here's a picture of a real aplysia and him holding one. So that's habituation. Basically, the response decreases as the stimulus is repeatedly presented. Another type of learning, non-association learning, is sensitization. And sensitization is sort of the opposite of habituation. It is the increase in a behavioral response to a harmless stimulus due to a preceding presentation of a different strong sensory stimulus. So for example, if you're walking alone in the park at night, if the lights suddenly all black out, then your response to a noise will be stronger than if the lights had stayed on, if nothing had happened with the lights, right? So in other words, some other stimulus can cause your response to a second stimulus to be much stronger than your response to either of the stimulus if they were independent. That's called sensitization. So for example, this happens in the California sea hare. You'll have a stronger contraction of the gill after a water jet if you shock the tail with electricity before it, right? So that's a sensitization. So there's two types of association learning. I should have this over here. So those were two types of non-associative learning, and now we're going to go over the two types of associative learning. So first is classical conditioning, which was Ivan Pavlov. Maybe you know him for his dog. He got the Nobel Prize for this experiment. And in classical conditioning, you pair two stimuli. One is the conditioned stimulus, which is usually not eliciting response. So dogs usually don't care about bells. But if you pair the bell with an unconditioned stimulus, like a food, a meat, which normally the dog has a response to, so it causes him to salivate because it smells good or it looks delicious, right? Then if you pair these together, then eventually the conditioned stimulus will directly cause the salivation, even if you don't have the unconditioned stimulus, even if you don't give the meat. So before conditioning, you have the conditioned stimulus, which is the bell, and it doesn't cause any salivation because normally bells don't cause dogs to salivate. And you have separately the unconditioned stimulus, which causes dogs to salivate. This is unconditioned response, is the salivation. Then during conditioning, you pair together the meat and the bell, and the dog salivates because of the meat, of course. But eventually, you can only give the bell, and the dog will salivate. The conditioned response will obtain. That's classical conditioning. Another type of association learning is called instrumental conditioning or operant conditioning sometimes. So in instrumental conditioning, a behavior is learned to result in a consequence, which is a reward or a punishment, the consequence. So if the consequence is rewarding, then the behavior will increase in frequency. And if the consequence is punishing, then usually the behavior will decrease in frequency. So for example, in this box, this is like maybe some type of Skinner box, where when this light goes on the left, then if the rat pushes this lever, then it gets some food, right? And so maybe when the light comes on, it will push the lever more. If when the light goes on, you add some electricity to the ground so it hurts the rat, then, but only if it pushes the lever, then the rat will no longer push the lever. Maybe it will stand over here and make sure it never touches the lever when the light is on because it knows it will cause a punishment. So Edward Thorndike was a psychologist who investigated instrumental conditioning by putting cats in what he called puzzle boxes. Here's a picture of one. The boxes were difficult to escape, so the cats had to find some different sort of levers to get out of the box. So the cat initially would make some random movements, but eventually it would, for example, push the lever by accident, and that would allow him to get out of the box, which for a cat is rewarding because he doesn't want to be in the box. And so this is called the law. Thorndike called this the law of effect, where pleasant consequences lead to higher probability of the behavior to be repeated. Of course, this can lead to sort of a cargo cult type behavior. So for example, if the cat noticed that he walked over here and then walked over here, and that let him out of the box, he might think that behavior of walking in this specific pattern caused, is what gets him out of the box. And it is maybe, but actually it's because when he walked over here, he stepped on the lever or something by accident, right? And so he sort of associated the wrong or a sort of a more specific behavior and not the actual sort of more functional mechanistic cause. That being that, even sort of more simple animals like cockroaches, bugs, can be conditioned. So for example, the cockroach can be conditioned using olfactory stimuli. So usually cockroaches don't like peppermint smell. They prefer vanilla smells. You can actually reverse this preference in cockroaches by giving peppermint, which is usually undesirable, with sucrose, sugar, which is reward, and giving vanilla smell with saline, which is salt water, which is kind of not a reward or a punishment. And by doing that repeatedly, you'll get the cockroach to walk towards smells of peppermint and walk away from smells of vanilla. Whereas normal situation is they walk towards vanilla and walk away from peppermint, right? So you can switch preferences of the cockroach via classical conditioning. Okay, so now the third part of this lecture, how can learning work in the brain? Well, we'll start with sort of just ideas. How would you think you would learn something in the brain? Well, will follow the way that people have historically thought. Well, to be able to learn, you maybe need to be able to represent what you need to learn in order to be able to associate it, right? So, for example, if you want to learn that snake should cause you to think danger, then you need to be able to represent snake and danger separately, and then somehow you can link them together, right? Via conditioning or whatever. But think about it. Do you have a cell in your eye, in your retina, that detects a snake? Probably not. Maybe some animals do. Maybe there's one in your brain that, via different shapes of light falling on your eyes says, oh, it represents snakes. And then how do you represent danger? Is there a danger neuron, a danger cell in your brain? Maybe you could just directly connect the eye cell to your leg muscles and run away. And actually some animals do that. Some worms or fish, like you directly connect some response in the eye to muscles on the other side of your body, and that makes you turn and run away. So it's a good idea. Let's think about how you could represent a concept. Well, Donald Hebb suggested in 1949 that memory concepts are represented as what he called a cell assembly, which is a distributed set of neurons connected by different connections. He called them neurites. And together these neurons represent a concept. So for example, habu, or snake, poisonous, might be represented in this set of neurons. And maybe you see a picture of only one part of a snake, and that activates only a few of those neurons, a few of those cells. But because the cells are connected, the activation spreads to the other cells of this neural assembly until the whole concept is active, and then you think of snake. This is called spreading activation. So maybe, of course, initially a hiss, just hearing a hiss, might not be strong enough to activate the other neurons and activate thinking of snake. Maybe because, of course, which makes sense, because other things hiss than snakes, vampires or something, I don't know. The tea kettle when you're making boiling water to make tea. And that's because these connections are not strong enough. Mechanistically, that's why it doesn't activate it. It might be the correct situation. You don't want only a hiss to activate a snake thought. But if you give a hiss and you have a picture of a snake, then it's enough to activate all these, all the other neurons, and you get the thought of a snake being activated. How do these become associated in the first place? How do you get the snake concept in the first place? How do these different parts of it link together so that they spread and activate each other? Well, Hebb also postulated that basically neurons that fire together, they wire together. So if you have things that are active at the same time, then the connection between them will strengthen. And if they're not active at the same time, the connection between them will weaken or get weaker. So if you repeatedly have a hiss and a snake picture, then the connection between them will strengthen. Until maybe afterwards the hiss by itself is enough to activate the snake picture and all the rest of the neurons, and that activates the whole snake concept. And you notice that repeatedly paired is kind of like, some type of association learning, classical conditioning or something, where you're associating two things that are normally not paired together. Of course, we want to make this a little bit more formal, so we will introduce the idea of a basic artificial neuron, or a basic neural network. So in a basic simplified model of a neuron, you have some neuron, some output neuron. So for example, I'll give it a concept later. And you have some input set. These are other neurons in the network that are connected to it. We'll call them A1 is activation of neuron 1, the activation, the response, how much it's excited. Activation of neuron 2, activation of neuron 3, activation of neuron 4. We'll call these input neurons because they're inputting to output neuron, OJ. They're connected with a certain weight, a certain strength of a connection. Weight 1J, weight 2J, weight 3J, weight 4J. And the output, the activation of this neuron J over here is just the sum of this activation times this weight. This activation times this weight. The dot product between the neuron activations and the weights. And so you can make a little bit more complex network with, for example, two output neurons representing snake and rabbit, or something else, like something else that's not a snake, right? And the input neurons maybe represent furry, small, snake-like motion and hiss. And we can imagine some starting condition, just for whatever reason, you have connections that have these weights. The weight between furry and snake is 0.1. The weight between furry and rabbit, appropriately, is probably 1.0. It's stronger, because rabbits are furry, but snakes aren't usually probably furry. And snakes have snake-like motions, so the weight is 1, but rabbits don't have snake-like motions, so the weight is only 0.1. So if you activate, for example, because you see something that's furry and something that's small, this activation will spread to along the connections, right? So 1.0 times 0.1 plus 1.0 times 0.5 is going to be 0.6. 0.1 plus 0.5 is going to be 0.6. And 1.0 times 1.0 is going to be 1, plus 1.0 times 0.5 is going to be 0.5, is going to be 1.5. So the activation, rabbit will be activated 1.5, energy level, whatever its arbitrary units, and snake will be activated only 0.6. So you say, oh, it's a rabbit, or the rabbit concept is more strongly activated than the snake concept when you have these inputs activated. Snake-like motion and hiss are not activated at all. They're 0, so they're not contributing anything, because 0 times 1 is still going to be 0, and 0 times these other weights is going to be 0, so they don't contribute anything in this situation. What if you only hear a hiss? Well, 0.5 times 1 is going to be 0.5. 0.5 times 1 is going to be 0.5, so snake and rabbit are both activated 0.5. The output is unclear. Of course, this is a weird, I don't usually think of rabbits when you hear a hiss, so maybe this is just, the baby was just, it's a baby's brain, it was just born, it doesn't know that, snakes hiss and rabbits don't yet. It has to learn it by associating it over time. Well, that's exactly what we're going to do. Hebbian learning is that the change in the weight is going to be somehow correlated with the activation of the input neuron times the activation of the output neuron. So the change of synaptic weight depends on the presynaptic neuron, the postsynaptic neuron's activation. So for example, if we activate both snake-like motion and hiss, then snake is activated 1.5, sort of strongly, whereas rabbit is only activated 0.6. So in this situation, if we sort of use Hebbian learning, these two are strongly connected. Hiss and snake are strongly activated at the same time, so maybe the weight of this connection will change more than the weight of this connection. And eventually the baby will learn that hisses go with snakes and hisses don't go with rabbits because they're not commonly co-occurring. So I'll go over this example. So let's say we have this activation for hiss over here. The change in weight of neuron 4 to snake, neuron output number 5, is going to be the activation of A4 times activation of output 5 minus, this is some what's called a bias term, -1, just some arbitrary term, in this case to make this example work. If you go into more actual learning rules like back propagation or something like that, you'll see that there's reasons for these numbers. So the change in weight is going to be 0.5 times, it'll be A4 times 0.5 minus 1, which is going to be, in this case, 1.5 minus 1, because there's 1.5, remember? And the activation of A was 1.0, so it's just 1. So the output is 0.5. So the change in weight is 0.5, positive 0.5. So the weight, the next time step, W45, this weight here, will be the weight at the previous time step, 0.5, plus the change in weight, which is 0.5 also. So the new weight is going to be 1. So after learning, you see it got thicker. We got a thicker weight here. The weight is strengthened because the hiss and the snake co-occurred. They were activated strongly at the same time. Meanwhile, this weight here between A4 and O6 is going to be the activation of 4, which is 1, times O6 minus 1 is going to be 0.6 minus 1 is negative 0.4. So it's going to be 0.6 minus 1 is going to be the change in weight, which is negative 0.4. So the original weight is 0.5. So we're going to do the change in weight plus the original weight is going to be the new weight. So it's 0.5 plus negative 0.4 is 0.5 minus 0.4 is going to be 0.1. So this weight 46 was weakened because the hiss and the rabbit concept, the O6, were not activated strongly at the same time. It was only activated a little bit weakly. So then after learning, you can already have the hiss. Now, when only the hiss is activated, snake is activated 1.0, so it's more strongly activated, whereas rabbit after this is now the weight's only 0.1, so rabbit's only activated 0.1. So rabbit is, the concept is less strongly activated. So you can understand this concept of how this is really abstract, sort of mathematical symbols, but you can see the concept of how you think that maybe neurons and concepts, cell assemblies in the brain being co-activated or not activated at the same time can cause different concepts to be associated and learned in the brain of an animal, of an organism. Of course, that's, like I said, very sort of abstract, mathematical, artificial, computational model. How does this relate to real brain cells, where there's actually different processes in brain cells that we know cause strengthening or weakening of connections between cells, between weakening or strengthening of synapses, and actually growing of new synapses or dying off of old unused synapses. So to review quickly how neurons communicate with one another, action potentials. So there's a cell here and there's a cell here, and there's a synapse here, of course, and this cell sends a signal along what's called an axon, or a connection between the cells. And this works by basically having an action potential, just quick inflow of sodium ions and outflow of potassium ions over a short period of a few milliseconds causes the membrane potential inside this portion of the axon to sharply increase. And that causes, by current flow, of course, the next sort of portion, the next segment, compartment of the axon to be also have an increased membrane potential and increased voltage relative to the outside the cell, which causes a voltage-gated sodium channels to open and causes an action potential, and that travels along the axon until it reaches a synapse at the end of the axon. And so here's the presynaptic, the input cell, and the postsynaptic, the output cell, right? And when it reaches the synapse, the voltage here increases, and that causes these voltage-gated calcium channels to be activated, which causes a cascade of different things that eventually causes vesicles to bind to the membrane and release their neurotransmitters, which are uptaken by receptors on the postsynaptic, the output neuron. Which causes a depolarization, increase in the voltage of the postsynaptic cell. So the postsynaptic cell gets activated if it's an excitatory neurotransmitter. Of course, as I'm focusing on excitatory neurotransmitters here, glutamate is the most common excitatory neurotransmitter in the central nervous system, in the brain. It binds to different receptor types on the postsynaptic, the output cell. So there's AMPA and kainate receptors, which are sort of basic ligand-gated receptors. And there's also NMDA, which is a voltage-gated receptor. And these, when you have glutamate binding to these, then it opens and allows sodium to flow into the cell, or in this case maybe calcium to flow into the cell, which causes the cell's membrane potential to increase, and the cell will become more activated. But that's not learning, that's just activating the cell. We want to know how cells can possibly, the strength of the synapse, for example, can increase. Well, this is called long-term potentiation, and this happens in a lot of different, it's one of the major types of learning in the brain. Here's an example from rat hippocampus. Even before LTP was discovered in the 1970s, we already knew that the mammal hippocampus was important for memory function. In the hippocampus, it's sort of like a nice little circuit where you have interrhinal cortex in the temporal lobe, feeds into a dentate gyrus, which is kind of the input to the hippocampus. So you have input, which then these cells in dentate gyrus go to actually CA3, and then CA3 cells connect to CA1 via Schaefer collateral connections. So basically, long-term potentiation strengthens synapses, and you can elicit long-term potentiation by, for example, stimulating the input axons to a CA1 neuron, so the blue and red lines in A. So these are the input connections to this neuron. So we're going to strengthen these connections, these synapses. So we're going to basically keep stimulating these with electricity to make this CA1 cell activated. And then we give a tetanic, a very, very high activation, basically simulating high activation to input 1, which is the blue subset of synapses over here. This fast stimulation leads to an action potential. It leads to this neuron being more activated, basically. And so in the future, when you activate input 1, input a CA1, this neuron will become more activated. This is graphed over here. Basically, you have a baseline response, and you give the tetanic stimulation, and then afterwards, the activation of this cell is stronger when you present the same input to these synapses. The opposite is, how does this work? So first of all, let's say, how does this work? Well, there's a bunch of different ways you can have it. Of course, we know that to activate the postsynaptic neuron, the CA1 cell here, you have glutamate binding to AMPA receptors, sort of normal, just ligand-gated receptors, that causes sodium to flow into the cell, and then that causes the postsynaptic cell to be activated normally. Its membrane potential will increase, depolarize. But also, actually, glutamate binds to NMDA receptors, which usually have a block. They're blocked by magnesium. But when the voltage of the postsynaptic cell is high enough, this block is removed, so like shown over here, which allows sodium and also calcium to flow into the cell from the via the NMDA receptors channel, which causes a further increase of the membrane potential of the cell. Also, there's other pathways that are activated by postsynaptic calcium ions inside the cell that cause things like, for example, new receptors to be embedded in the postsynaptic membrane, which will make it more sensitive. It'll be able to basically get more sodium more quickly in the future. Another thing that can happen is you can actually have kinases, so calcium can activate some kinases in the cell, which will phosphorylate the AMPA receptors and make them sort of more efficient. They'll be able to better let ions through, and that will also increase the effectiveness, the efficacy, the strength of this synapse. The opposite of LTP is LTD, long-term depression. And this is basically what's shown here on the, so this is the unstimulated. So this is sort of the control over here, right? We didn't stimulate these inputs to this neuron repeatedly, and so they were in the original, the LTP. They stayed the same. Even if we stimulate these with the same response, the output of the output in the cell is the same. For long-term depression, it's basically the opposite. In this case, we give a very, very weak stimulus to the input 1, and it doesn't cause the cell to fire. So basically these have very low inputs, and this is very, 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 very low activation, and that causes a decrease in LTD, a decrease in the response given the same future input to the input 1. The control is input 2. If we don't give any input there, they stay the same strength basically. So this is LTD, long-term depression. And this can happen in a couple of different ways. One way to elicit is, of course, low-frequency stimulation, which leads to calcium influx in the postsynaptic cell, but only in very small amounts, which leads to protein phosphates, phosphatases that actually remove phosphate groups from the AMPA receptors and reduce their ability to let ions through. So the AMPA receptors leads to the AMPA receptors are less strong, or AMPA receptors are removed from the membrane, and that weakens the synapse. Okay, so summary, LTP and LTD. So LTP strengthens the synapse when the neurons fire together, or specifically when a postsynaptic action potential follows an EPSP caused by a presynaptic action potential. It seems that it strengthens. So that's called synaptic spike timing dependent plasticity. I don't talk about that in more detail here, but that is one other sort of type of learning that maybe is a type of LTP. The opposite, LTD, weakens the synapse when a presynaptic action potential does not cause a postsynaptic action potential. Or, in other words, the postsynaptic cell is not activated, but the presynaptic cell is. Okay, so summary of today, what we did, we covered different form of learning, especially non-declarative memory. So I covered the different sort of psychological separations of types of memory or learning. So we talked about non-declarative memory versus declarative memory, versus and non-associative learning versus association learning. And within these, we covered habituation and sensitization. We covered classical conditioning and instrumental conditioning as two types of associative learning. Then for associative learning, we learned about how that could happen in the brain abstractly. We learned about Hebb's idea that concepts or thoughts, or whatever you want to call it, are represented in the brain via cell assemblies. And the modification of connections is via learning, via co-activation of different of the cell assemblies or parts of the assemblies can make them become associated with one another. To understand this, we had to learn about neural networks, artificial neural networks, and how you can have basic type of Hebbian learning inside of them to, for example, associate hiss with snake, but not with rabbit. And finally, I covered how that could, some of the mechanisms, I overviewed them very briefly, how that type of Hebbian learning could be implemented in the brain, specifically as long-term potentiation, LTP, and long-term depression, LTD, which operate via the action potentials in the pre- and postsynaptic cells, and different calcium signalings in the postsynaptic cell embed or remove AMPA receptors, for example, in the postsynaptic cell's membrane, and also attach phospholates to the AMPA receptors to make them more or less effective, thus weakening or strengthening the overall strength of the synapse, similar to weakening or strengthening the strength of a connection in our abstract artificial neural network model. So that's all for this week. I will upload the quiz for this week, and I guess one for last week as well about motivation sometime this weekend, and I'll email you and tell you where to find it, so don't worry about that if you haven't received an email from me yet.